All right, so here we go with the lymphatic system. Again, a lot of times it's kind of thought of as synonymous with the immune system, but each of these systems tie into a lot of structures that are involved with other organ systems or other tissues as well. So when we're talking anatomy and physiology, we're talking structure and function. So we talk about the fluid, the lymph fluid. We're talking about extracellular fluid, interstitial fluid, stuff that's not inside blood vessels and not inside cells. We also have vessels that need to transport all of that. They're going to be valve-based vessels, which are similar to veins, and they're going to pump through muscle contraction instead of being pumped through the heart. And then we also have lymphoid organs like lymph nodes, spleen, thymus, tonsils, pars, patches. And then as we get talking about those, we really need to discuss the white blood cells. So um, here we go with our lymphatic system. Again, our structures that we're talking about. We're talking about our tonsils up here, our cervical lymph nodes, our thymus, our axillary lymph nodes, our spleen, our inguinal lymph nodes, bone marrow, Peyer's patches, but we'll look at those in a little more detail. Again, the big part of this, like I can give you just a general PowerPoint or whatever, but it's important to visualize. And this is again the whole idea of science is to get involved with models to try and make complex ideas easier to understand. So the first thing I want to look at, short video, but just kind of getting a concept of these vessels.
The lymphatic system is a network of vessels lying adjacent to the capillaries. It aids the circulatory system in transporting fluids and is a vital part of the immune system. In and around the cells of the body, extracellular fluid collects waste products and carries nutrients and oxygen to the cells. Some of the fluid travels through the capillaries. Some, especially if it contains proteins too large to enter the capillary membranes, circulates through the lymphatic vessels. The lymphatic vessels flow through several lymph nodes. These lymph nodes contain concentrations of white blood cells which kill bacteria and other harmful agents trapped in the nodes. The tissue fluid returns to the circulatory system through the large veins near the heart.
So there we go. Again, adjacent to all the blood vessels, we have lymph vessels. So one way to picture this, again, as we're looking at this, is that they're all intertwined. A lot of times we think of the arteries and the veins and the capillary loops in between there, but the reality is in all this tissue area, we also have these lymph vessels that are circulating all in there. Now, why in the heck don't we see lymph vessels in most models? Part of it is to make things simpler. Like you leave them out so then you can see other stuff in the model that you want to show. And you see that here when we did our little integumentary system model. I have these capillary loops and I have blood vessels and everything like that. But we're also going to have lymph. We're going to have extracellular fluid in this space. So you're going to have lymph vessels that are also going to transport that fluid and move it ultimately, clean it up, and then move it back into the bloodstream. So if we're going to talk about lymphocytes or cells of the lymphatic system, we need to talk about all these white blood cells. And where do white blood cells come from? They come from bone marrow because that's where blood cells are made. Now, we start with a basic stem cell that can differentiate into all these different types. And again, we're focused on not just the lymphocytes, the B's and the T cells and the natural killer cells, but we also get into neutrophils and basophils and eosinophils and macrophages, monocytes, all that kind of business as well. And red blood cells also originate, if you trace them all the way back, to the same types of stem cells. Again, when we see this, we see, okay, we have this stem cell that can be turned into many different types of blood cells. And when we look at this chart, we're like, oh, red blood cells, erythrocytes, those are a tiny little portion of this. And our thrombocytes are our platelets, just like sloughed off pieces of these megakaryocytes. But we have like, oh my gosh, all these different types of white blood cells. And again, in that inflammatory and immune response video, you get worked into that vocabulary a lot. But it's easy when you look at this to think like there's so many more white blood cells than red blood cells. The reality is, when we look at this and it's broken down, if all of our blood is this, and it starts getting broken down smaller and smaller and smaller, there are a lot more red blood cells than there are white blood cells. And again, our white blood cells, we're getting to our neutrophils, our lymphocytes, which are our B and T cells. But our monocytes, our eosinophils, and our basophils, which are real similar to some other like mast cells here. So let's start talking about structures in particular. Again, our bone marrow is where all of these lymphocytes originate from, and all the different white blood cells are leukocytes. But we also have this thymus, and the thymus is kind of unique in that it shrinks over time, but it's really important. The general function is that it matures T cells. We have helper T cells and we have killer T cells. So from the bone marrow, some of these lymphocytes go to the thymus, and we call those thymocytes or T cells. Now, our helper T cells have these CD4 proteins on their surface, which interact with the MHC2 proteins, which are presenting peptides that are from on macrophages. So helper Ts are specifically designed with the proteins that are on their surface to interact with macrophages. Whereas our killer T cells, they have a different type of protein on their surface, and they're going to interact with these MHC1 proteins. That those proteins are presenting peptides that exist on every single cell in our body. So our killer Ts are monitoring everybody, and our helper Ts are only focused on interacting with macrophages and then ultimately B cells once they've been activated. So what the heck does maturing T cells actually mean? And this thymus organ, it gets much, much smaller as you age, and it becomes, less, plays less of a role. But when we mature T cells, ultimately what that's meaning is that these helper Ts and killer Ts, through a kind of a complex process, they ensure that the reaction that they react with foreign antigens but do not react with self antigens. So if there are duds in this process, if you have some T cells that are initiating an immune response against ourself and causing like a disease where our immune system is attacking our own body, then at this point we're hopefully having this checkpoint where those go through apoptosis and are destroyed because they're not working right. So it's through this like checkpoint in the thymus to make sure that the T cells are interacting and will react when foreign antigens are present, but will not react when self antigens are like knowing who we are. So we don't like initiate an immune response against autoimmune disease, that kind of stuff. All right, so moving on. Now we have all these other areas that are homes for lymphocytes and are meant to clean things up or activate the immune system. And the first thing we're going to interact with is air or food that's coming into our body. And our tonsils and our adenoids are involved with that process. So again, most of the time you can look in the mirror when you're sick and you can see big tonsils, and they'll check your throat, see if they're engorged. So those tonsils, they have certain types of cells on their surface which can capture foreign material and activate lymphocytes. And again, if you activate those lymphocytes, you're ultimately going to proliferate or make more of them to initiate a activated immune response, a specific immune response. So Peyer's patches work very similar to tonsil tissue, and they have both of these have these M cells here, and underneath those M cells, the M cells kind of do the catching, and then we have B cell activation, and we can get the immune system kicked off here with with our helper T's and our dendritic cells, which ultimately do act a lot like macrophages and being activated and presenting antigens. So where are our Peyer's patches? Well, if we follow our digestive tract here from the esophagus into the stomach, we get into the duodenum, which is this first section of the small intestine. And that area of the small intestine is still getting bile secreted in and juices from the pancreas secreted in, and itself, it secretes some digestive enzymes. So we're still breaking stuff down in the duodenum. We're not really absorbing much. From the duodenum, we head into the jejunum, and there's where most of the absorption is going to take place. And the Peyer's patches, these collection of lymphatic tissue, is going to exist mostly at the end of the jejunum and the ileum. The ileum is the last section of our small intestine before it dumps into the colon or the large intestine. And then we work it around here and then ultimately head on out. So we'll talk about other areas of your body where you have places that are monitoring for non-self protein or non-self antigens throughout your body, and not just here, but with our tonsils and our Peyer's patches, there's like a higher concentration of these things. So lymph nodes also, if we're filtering or monitoring our air and the food in our digestive tract, we're also going to filter our lymph. So from those lymph vessels, we have lymph nodes many places in our body, but we typically think the largest concentrations are in our neck, which are our cervical lymph nodes, in our armpit, which are our axillary lymph nodes, and in our inguinal region, or kind of our groin region. And again, as the lymph comes in, it kind of is going to pool and slow down here because it has so many things coming in and then only one small outlet. So it's got to slow down in order to make it through all of this reticular fiber. And then you have all these lymphocytes embedded within that tissue. So again, we are looking for anything foreign that could be in our system that could initiate an immune response and ultimately clean up all that extracellular interstitial fluid prior to dumping it back into the blood. We also have our spleen. And our spleen, once we get into the blood, the spleen is going to be more involved with actually filtering the blood. And it's the same kind of idea. It's the same basic kind of tissue that we see with our lymph nodes. And in addition to checking in for foreign antigens and activating lymphocytes, B cells and T cells, the spleen is also really in charge of recycling old red blood cells and breaking them down. So the spleen is a huge storage facility too of red blood cells and lymphocytes. But the main idea behind your spleen is that it's filtering blood. And again, activating lymphocytes is part of our immune system, specific immune system. So where else? Again, those are the big ones as far as structures of the lymphatic system go. But if we can remember back to this, we've got our Langerhans cells within our epidermis. So it's not like these are the structures I showed before are the only places where we can activate the immune system. A lot of this can happen in many different locations. And again, if we're talking about this, we just did our inflammatory and immune response. And where would this guy be? It'd be buried into this epidermis here. And in addition to having our macrophage activate the immune response, we can also get that going on with our Langerhans cells. Another place, we also have these Kupffer cells within our liver. So again, these are white blood cells. These are dendritic cells that are meant to activate the immune response, and they are in our liver. And part of that, liver is an amazing organ, but all of this blood from our digestive tract ultimately first heads to our liver prior to heading to our heart. So that's a good place to, check for foreign material. In addition to our Peyer's patches in our small intestine, we also, have other places around our body that are monitoring foreign antigens and trying to get rid of things that don't belong in our body. Again, those are all our structures, and I kind of hit up on the functions as we were going. So ultimately, the functions of the lymphatic system are to produce, maintain, and distribute lymphocytes, B cells, and T cells. And we're going to serve into this whole immune system, like we're going to get into all of our other leukocytes too, our white blood cells. Now, all that fluid and solutes from other tissues, we're going to return all that to the blood and as a result of that, we're also, from those other tissues, we're also going to be able to distribute hormones, nutrient, and waste products back to general circulation. So I want to quick run through these leukocytes, but we spent so much time with that inflammatory and immune response slideshow where we hit on most of these. So I don't want to spend too much time, but I'll hit them back through again. These are white blood cells involved with an inflammatory response, sending out cytokines to begin communicating inside the body. We've got mast cells, we've got neutrophils, we've got macrophages. And again, we can look at them in this type of a chart, which I showed you with the other video. But as we run through this really quick, this is the model that I've created. It's one way to look at all of their functions as a part of a story instead of just into a chart. So quickly here, our body recognizes damage, and it doesn't have to be, because we think about like viruses and stuff that are airborne, they're causing cellular damage too. And that's not necessarily like getting stabbed by anything and bursting cells in that way, but they can still damage cells and cause cells to burst, which can initiate an inflammatory and immune response that way. But we have foreign material, pathogen present. We have damaged cells that causes mast cells to release histamine, which causes dilation in blood vessels, capillaries to get leaky, which allow neutrophils to come in as bacteria slayers to engulf and destroy the pathogen. As we begin our specific immune response, we have macrophages, which are going to engulf and destroy pathogen. And then on their MHC2 proteins, they will present foreign antigens. And that's when we would say that a macrophage is activated. A helper T cell is going to come in contact with that foreign antigen, and it's going to send out cytokines to call in the lymphocytes, the other lymphocytes, for more assistance. The killer T cell's job here is to monitor our own cells for infection. It also will monitor for cancer, but when it finds foreign particles on our MHC1 or abnormal peptides presented on our MHC1 proteins, it will destroy cells before those cells can burst and spread the infection and spread all the replicated copies. Our B cells get involved. They need to come in contact with the pathogen themselves, and then they will present the foreign antigen. When they come in contact with the activated helper T cell, we can proliferate or make many more copies, go through mitosis of our B cells and have them become plasma cells, which ultimately secrete tremendous amounts of antibody proteins. Those antibody proteins neutralize agglutinate, they clump, they make them unable to hurt us anymore, those pathogens, and also mark them so that eosinophils can come in and destroy them more easily. With this process, we end up with some memory cells so that in the future, we can skip many of those intermediate steps and go directly to mass producing antibodies. And that is kind of the idea behind vaccines. So again, that's running through all of that kind of stuff. Now, cell-mediated immunity, to look at these models in another way and get in on these killer T cells a little bit more. When we look at these our cells all have these MHC1 proteins on their surface as markers. Now, as our cells go through normal metabolism, they are going to recycle some proteins, some peptides, and put them on these MHC1 proteins. Our killer T cells are going to go around and check all of these. And if it looks normal, it's going to leave it alone. However, if it's cancer or whether it's infected, there might be some abnormal peptides being presented on the MHC1 proteins. If that's the case, the killer T cells will then secrete perforin, which can poke holes. And if you poke a hole in its lipid bilayer and dissolve that, it's going to cause cell death. We can also send out cytokines, which again are like hormonal messengers, which would go and talk to the DNA and start turning on genes to that would cause apoptosis or cell death. Now, one of the types of proteins that can be made or genes that can be turned on are DNA ACEs. ACEs are enzymes, and what are they actually breaking down? They're breaking down the DNA. So within the genome, there are genes to make enzymes to self-destruct, and we talked about that a little bit with our tumor suppressor genes and causing and destroying cells. So killer T's just kind of kick that on or just secrete perforin that'll perforate the cell membrane and destroy it. Natural killer cells, something we don't talk about all that much, work very similar in that process, but the theory is that they are more looking for missing self as opposed to just foreign. So if a normal cell has a certain density of MHC1 proteins, natural killer cells are thought to possibly find them because um, there's they have less density of MHC1 or an abnormal density of MHC1 proteins, which even though the peptides presented on them may be normal, if they don't have the right amount in the first place, there might be some type of mutation or mess up with this DNA, and that should also elicit killing this cell. And that's the whole big story. You guys can go back through the PowerPoint and check all that kind of stuff out, but that's basically what I just explained. Now, interesting things here. As far as fighting cancer, I know we're so focused on COVID-19 and everything like that, but it is pretty interesting to think about some of the new cancer research, which ties into not just fighting it with chemotherapy drugs or radiation or surgery, but actually training your immune system to go and find that cancer. So I calling them MHC1, this calls them MR1 proteins, but T cells interact with that. And what they're saying is that they are trying to kind of re take those cells out of the body and kind of act like a little bit of like a thymus would, where they kind of train and modify the T cells to find the cancer that they have missed. So if cancer is spreading, what that ultimately means is that um, these killer T cells did not actually recognize it as foreign. So um, if they go undetected, the cancer can spread. If it finds it as cancer and messed up, not just infected cells, but if they're messed up cancer cells, they should get destroyed. And your killer T cells are another checkpoint kind of in preventing cancer. So um, how they have some of this stuff that works for leukemia, and I actually have a cousin who's doing some of this research out east, but they take some blood out and then they take out the T cells and then they actually genetically modify the individual T cells based off of that person's cancer type. So then they ultimately end up growing their T cells, grow receptors to specifically hunt out the specific types of cancer that they have, and then they can go through mitosis and duplicate them in the lab, and then they actually put these genetically modified T cells that have been trained up to recognize the specific type of cancer back into the patient, and then they actually go and out and search out and kill the cancer, which is just amazing, you know. And again, this is a very early part of the development of this treatment, but it's pretty exciting for sure. So again, that gets into killer T cells and not just like viral or bacterial infection, but also how your immune system does monitor and hunt out cancer as well. So now getting into these other types of surface markers, when we start talking about antibody-mediated immune response, we start talking about the MHC2 proteins. Now, MHC2 proteins only exist on macrophages. So again, a macrophage is a human cell, so it's going to have MHC1s, and its MHC1s are going to present normal because this is a normal, healthy cell. However, on its MHC2 proteins, it's going to present, when it's activated and has engulfed and destroyed a pathogen, a foreign antigen from whatever it destroyed. When it happens there, the helper T will get involved here, and again, cytokines will be sent out, call in the B cell. The B cell will interact, and again, it'll present the antigen. The helper T will go from the macrophage, and once it's activated, it will go and then interact in direct contact with the B cell and send out cytokines, which will then cause that B cell to become a plasma cell, which mass produces antibodies. And those antibodies will then be sent out, these proteins will be sent out to go and clump and agglutinate and neutralize so they can't infect us, all that pathogen that's in our system. So again, that's a summary of all that kind of business that I just explained. But again, explaining all this kind of stuff, sometimes it helps to see an actual animation. So we're going to take a look at one of those now.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
All right. So as we get into that, again, that's all of our cell-mediated immune response and antibody-mediated immune response, how our B cells and T cells work, and helper Ts and killer Ts and plasma cells and memory cells and all of that kind of stuff. Sometimes it just helps to look at another model of that to understand activation. When we say something's activated, what does that actually mean? Now, I think it's important to also understand how viruses work. I know we're doing a lot of reading about the coronavirus. but I think this animation is pretty helpful too, especially when it comes to retrovirus. And let's take a look at HIV.
When HIV first enters the human bloodstream, the virus circulates throughout the body. The protective surface of HIV is studded with particular proteins called GP120, and inside is a capsid that contains the viral RNA and viral enzymes. Initially, the GP120 protein on the virus is only able to bind to a cell surface protein on the macrophage called the CD4 receptor.
Now, again, like a virus doesn't necessarily infect or embed itself into every single cell of our body. There's only specific viruses that target like our respiratory tract. Okay, so it's not, and then there's other ones, that are focused that infect our liver. And the HIV virus is focused on actually attacking our immune cells.
A second receptor protein, CCR5, must also be present on the macrophage for the virus to enter the cell by endocytosis. Viral RNA and enzymes are released into the cell's cytoplasm. Reverse transcriptase uses the viral RNA to synthesize first a strand of viral DNA and then the complementary DNA strand. The viral DNA then moves to the nucleus of the cell, where it integrates into the host cell's DNA.
So a lot of viruses, especially like our coronavirus, it's not a retrovirus. It doesn't turn itself back into DNA. It just uses the RNA that's injected into the host cell, and that RNA goes straight into the ribosome and starts cranking out all the proteins in order to remake that same virus. But retroviruses actually have this reverse transcriptase, because transcription is DNA to RNA. So reverse transcription, and the ACE on the end means it's an enzyme which helps with chemical reactions, actually turns it back into a DNA strand that can become permanently part of our DNA.
Transcription of the DNA within the nucleus now results in the production of viral RNA. This viral RNA can serve as the genome for new viruses and can be used as messenger RNA to produce viral proteins during translation by the ribosomes. Complete viruses are assembled and released from the cell via exocytosis, which causes little harm to the macrophage.
Again, a virus can't replicate on its own. It has to hijack other cells and use their machinery, their ribosomes, to actually make copies of themselves. So again, we don't consider viruses necessarily alive because they're not maintaining their own homeostasis, and they just hijack our host cells.
HIV cycles through macrophages over a period of years and continues to multiply while doing little apparent damage to the body. Eventually, the gene that codes for the GP120 protein is altered by mutation. The altered GP120 protein changes its co-receptor allegiance and now binds to a different co-receptor, CXCR4, which is found on the surface of CD4 plus T cells.
So the CD4 receptor and those CD4 cells, these are helper T cells, okay? Because the CD8 are the killer T cells. So ultimately, the first infection of the HIV virus embeds itself into macrophages and uses macrophages to make copies. And it doesn't hurt us until it becomes mutated, but then once it becomes mutated, it stops hijacking macrophages and instead starts hijacking helper T cells.
The same processes occur within the T cells, resulting in new virus particles. However, as these new viruses leave the T cell, they rupture the plasma membrane, which kills the cell.
So there we go. I mean, if the viruses, when they're secreted, now different viruses harm your cells in different ways. Sometimes they mess with your DNA and it causes all the cells to ultimately go through apoptosis and destroy themselves. Sometimes they just hijack so much of the ribosomes and everything that those cells that they're infecting can't carry out their normal functions because they're so busy making copies of the virus. And then again, this way, a lot of times when the viruses make all these copies, when those viruses finally burst the cell and spread to other cells, they kill the cells, and when you start killing cells, you start getting sick. Now, in this case with HIV, if you start destroying, now think about this, if you start destroying all the helper T cells, if you interrupt that section of the immune response, you don't have helper Ts, that means you can't activate and turn the B cells into plasma cells. If you can't get plasma cells, now you can't make antibodies. And if you can't make antibodies, very simple things can kill you. So when we say AIDS is acquired immunodeficiency syndrome, that immunodeficiency, it's not the virus that kills you necessarily. You end up dying from other types of um, diseases that because your immune system is compromised, because the virus, the HIV virus, when it's mutated, ultimately destroys all your helper T cells.
As these newly released viruses invade and destroy other T cells, the body's immune response is weakened, and this leads directly to the onset of AIDS.
Okay, so this is quite a long video as it is. One last thing I want to talk about is vaccines. So when we start looking at the lifespan of white blood cells, they are quite short, right? So we need to have our bone marrow continue to making these new things, but memory cells do have the ability to live much, 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 much longer than some of these other white blood cells. So with that ability for them to go through mitosis and to live a much longer period of time and not get destroyed themselves, that is the idea of vaccines. And a lot of the idea behind vaccines originates with Louis Pasteur. And I read this book when I was a little kid and made me scared of needles. But it's all about rabies. And my sons have read this book too. We have it at our house. And they're talking about in this book, this is my introduction to vaccines. And when it comes to this, a lot of times the book explained it like he was shooting in these little soldiers in order to fight off the rabies virus that had infected this child. But the reality is, you're not shooting in the good guys, you're shooting in weakened or dead versions of the bad guys. And those weakened and dead versions of the bad guys ultimately trigger that production of memory cells. And without them, they themselves getting you sick. So that's the whole idea behind vaccines. And when he was starting to first come up with this, it's a little bit of like scary time in medicine, because there was a lot of this before them that they didn't even realize that they should wash their hands before performing surgery. The germ theory of disease hadn't fully taken hold yet. So like people are dying because they're getting surgery, not surviving because of the surgery. And this story is really interesting when you get into the reality of it. And there's a lot of his Pasteur's logs, his journals that were not released until just fairly recently, even though this was happening in 1885. It was kind of like passed down through his family and they didn't make anything public. Because he had done a couple trials with dogs and stuff, but he didn't really know if this was working. And then he took a child and started injecting them with inoculations, or smaller and smaller, and actually small versions of the virus, and then larger and larger versions until it developed immunity. And this boy survived, and he was a hero. If it wouldn't have worked, he probably would not be the hero that he was. But we have so much advancement in medicine because of it. But there are other things that that didn't quite make the news that maybe other shadier businesses were taking place. But anyways, it's a really interesting topic to look into. There's a lot of good videos out there about immunity and how vaccinations work, but I feel like this video is long enough, so I'm going to cut it right there. So hopefully that's helpful to understand a little bit more about the lymphatic system.